I'm SAKINAH.
Hey, I'm IRFAN.
Hi, I'm SAMHI.
Hi, I'm SYAKIRAH.
Call me NATASHA.
Sekarang kami berada di Taman Botani Putrajaya. Let's go!
Hi everyone, I'm SYAKIRAH. For practical 1, our objective is to calculate and describe the mean, variance and standard deviation of leaf measurement. We also aim to understand the concept of correlation between two variables and determine the relationship between leaf length and leaf width, whether it is linear or non-linear. For the methodology, we first collected 10 leaves from the same tree species. Then, next, we measure the length and width of each leaf using a ruler. After collecting the measurement, we calculated the mean, variance and standard deviation to analyze the variation in leaf size. The data were then recorded and tabulated for further analysis. We repeated the same procedure for 2 more tree species, giving us a total of 30 leaves from 3 different species for comparison. This method allowed us to compare leaf characteristics among different species and evaluate the relationship between leaf length and width.
Assalamualaikum, my name is Muhammad IRFAN. So I will be presenting the answer for post-lab question 1 and question 2 from practical 1. Basic concept of statistic and variation. So the first question is to discuss the differences of or similarity of the mean of length between different set of leaf. Do the same for the mean of width. And the second question is what is meant by variance. The leaf have different measurement of width and length. What is the meaning of this statement? Discuss with respect to variance. What is meant by standard deviation? The first question asks us to discuss the differences or similarity of the mean or of length between different set of leaf. Do the same for the mean of width. First, let's look at the mean leaf length. The results show that set 1 has a mean length of 22.98 cm. Set 2 has mean length of 18.37 cm. And set 3 has the highest mean length of 30.08 cm. From this value, we can clearly see that the average leaf length is different for each set. Set 3 has the longest leaf, which means that on average, its leaves are much larger than those in set 1 and set 2. Set 2 has the shortest leaf, while the set 1 falls in between the two. Now, let's look at the mean leaf width. The average width of the leaf in set 1 is 7.40 cm. Set 2 is 6.99 cm. And set 3 has the highest average width of 15.44 cm. Unlike set 3, set 1 and 2 have very similar mean width. The differences between them is only 0.41 cm, which is relatively small. This means that the average width of the leaf in set 1 and 2 is almost the same. However, set 3 has the much larger mean width, more than twice that of the other two offset. Overall, we can conclude that set 3 contains the largest leaf because it has the highest mean value for both length and width. Set 2 contains the smallest leaf, while set 1 represents medium-sized leaf. Therefore, the ranking of leaf size is set 3 followed by set 1 and finally set 2. So, why the average leaf size are different? So, there are several possible reasons why the average leaf size are different. So, one of the reasons is genetic variation, where different plants naturally produce leaves of different size. Another reason is environmental factors such as sunlight exposure, water availability, soil nutrient, temperature and growing condition. In addition, the age of leaf may also affect their size because older leaves usually have more time to grow. Therefore, comparing the mean value helps us understand the overall characteristic of each set and identify which group contain larger or small leaf. The second question asks us to explain the meaning of variant. The statement the leaf have different measurement of width and length, and the meaning of standard deviation. Firstly, variant is a statistical measure that tells us how much the data are spread out from the mean. In simple words, variance measure how different the individual values are from the average value. If the variance is small, it means that most of the measurements are close to the mean, so the data are more consistent. On the other hand, if the variance is large, it means that the measurements are spread out over a wider range and vary greatly from one another. In this experiment, the variant values show clear differences between the three sets. Set 1 has a leaf length variant of 17.14 and width variant of 5.88. Set 2 has the lowest variant with 7.99 for length and 1.39 for width. Meanwhile, Set 3 has the highest variant with 55.17 for length and 21.14 for width. This result tells us that the leaf measurement in Set 3 vary much more than the other two sets. Some leaves are much larger than average, while the others are much smaller. In contrast, the leaves in Set 2 are more uniform because their measurements are closer to the mean. The statement the leaves have different measurement of width and length simply means that not every leaf has exactly the same dimension. Some leaves are longer, some are shorter, some are wider, and some are narrower. This is known that as a natural variation, and variant is the statistical tool that helps us measure the variation quantitatively. Next is standard deviation. Standard deviation is the square root of the variance. It tells us the average distance of the measurement from the mean. Unlike variance, standard deviation is expressed in the same unit as the original data, which in this experiment is centimeter. This makes it easier to understand and interpret. Looking at our result, Set 1 has a standard deviation of 4.14 cm for length and 2.43 cm for width. Set 2 has the lowest standard deviation with 2.83 cm for length and 1.18 cm for width. Again, Set 3 has the highest standard deviation, which is 7.43 cm for length and 4.60 cm for width. This value confirms what we observe from the variant. The leaf in Set 3 show the greatest variability, while the leaf in Set 2 are the most consistent in size. For the conclusion, question 1 and 2, the mean is used to describe the average size of leaf, while variance and the standard deviation describe how much the measurement vary around that average. From our result, Set 3 has the largest average leaf size and the greatest variation, indicating that its leaves differ considerably from one another. Set 2 has the smallest average leaf size and the least variation, showing that its leaves are more uniform. Set 1 falls between the two sets for both average size and variability. Overall, these statistical measures are important because they help us analyze biological data accurately, compare different groups of samples, and better understanding the nature variation that exists among living organisms.
Hi, Assalamualaikum. My name is NATASHA HUDA. Next slide is practical 1, question 3. Why is it necessary to have different sets of measurement for the measurement of leaf width and length? Different sets of measurement are necessary to ensure that the data collected are accurate, reliable, and representative of the leaf population. Individual leaves naturally vary in length and width due to factors such as age, position on the plant, exposure to sunlight, and the availability of water and nutrients. Measuring several leaf provides a more accurate estimate of the average leaf size and reduce the effect of natural biological variation. In addition, taking multiple measurements minimize the impact of random measurement error and improve the reliability of the result. It also allows statistical analysis such as calculating the mean and standard deviation, which help describe the average leaf dimension and the variation among samples.
Kita nak buat kuadrat and petik lah. Jom. Let's go.
Today we gonna do quadrat sampling. So the objective of the quadrat sampling is to observe, identify and to record the distribution and abundance of plant species within the selected area and to determine the factor affecting their distribution.
So we already setting up the quadrat. The size of quadrat is 6 and 6 meters square. So as we can see, we already tied a rope around the edge. So now we will count the number of species within the quadrat. Okay, let's go.
Okay, hello everyone. This species tree is Paricia insignis. In Malay, Sepul Beludu. Like we can see, this tree is quite tall and its leaf quite wide.
Second species, this species is Syngium species. The family name is Miraceae. The normal common name is Kelat. It's from Southeast Malaysia. This species spanning from Africa and Madagascar through South and Southeast Asia, such as Indonesia, Malaysia, to Australia and the Pacific.
For third species, we call it Hopia odorata, and the family name is Dipterocarpaceae. Okay, for in Malay name, we call it Merawan siput jantan. So for the description for this species is you can see the tall evergreen tree. So the related native to South and South Asia and including countries like Bangladesh, Colombia, India, Laos, Malaysia, Myanmar, Thailand and Vietnam also. So the height of the tree is 45 meters.
The plant species that we found in this quadrat, we record a total of 2 individuals. For this species, we name it as species A. The plant species is tall and slender in appearance. Based on the observation, this species show vertical growth with relative thin stems compared to other plants in this quadrat.
We call it the species B. So this species we have 3 individuals in our quadrat. So our character this species is they have like sharp spine, and also the large fan-shaped leaf. So as you can see, the spine itself is as a defense from again herbivore.
Assalamualaikum. My name is AHMAD SAMHI. Today, I will be presenting the Puslet question for Practical 2. Factors that affect the distribution and abundance of organism. I will be answering question 1 and question 2. Syakirah will present question 3, while question 4 will be discussed by Sakinah. Lastly, for question 5 will be presented by Natasha. For question 1, define the term quadrat in practice. A quadrat is a square or rectangular frame with known dimension that is placed on the ground to study and sample organism in a specific area. It is commonly used in ecological studies to estimate the abundance, density, frequency, and distribution of plants or other organisms that do not move. In this practical, a quadrat measuring 6 meter by 6 meter was used to define a fixed sampling area. All plant species inside the quadrat were identified, counted, and recorded. This method helps researchers estimate population size and population density in a study area. It is useful because it provides a systematic and standardized way of collecting ecological data, so results can be compared between different sites. Next, for question 2. What are the two basic requirements of all quadrat techniques? The first requirement is that the quadrat must be placed randomly within the study area. This is important to minimize sampling bias and to ensure that every part of the habitat has an equal probability of being selected. By using random placement, the data collected becomes more reliable and truly representative of the overall ecosystem, rather than being influenced by the observer's selection of specific area. The second requirement is that the organism within the quadrat must be clearly identifiable and countable. This ensures that accurate data can be collected during sampling. Quadrat methods are most suitable for plants and other sedentary organisms because they remain stationary during the study period. This allows researchers to count individuals precisely within the defined area. In contrast, highly mobile organisms such as monkey, insects, or birds are not suitable for quadrat sampling, as their movement in and out of the sampling area can lead to inaccurate or inconsistent counts.
Hi everyone, I will present the advantage and disadvantage of using a larger and smaller quadrat when surveying sedentary organisms such as plants. First, let's look at the larger quadrat. A larger quadrat has several advantages. Firstly, it covers a larger study area, allowing us to sample more of the habitat. Secondly, it includes more organisms and species, making it easier to record a greater number of individuals. Third, because it samples a larger area, there is a higher chance of finding rare species. Another advantage is that it reduces sampling error because the results are less affected by random variation. Finally, a larger quadrat provides more accurate estimate of population density and species diversity, giving a better representation of the habitat. However, there are also some disadvantages. A larger quadrat is more time consuming because it takes longer to complete the sampling process. It also requires more effort as more organisms need to be counted and identified. In addition, it is more difficult to set up, especially in dense vegetation or uneven ground. It may also require more equipment, manpower, and higher costs. Now, let's move on the smaller quadrat. A smaller quadrat is much easier to use. It is quick and easy to set up, making it convenient for fieldwork. Sampling can also be completed more quickly, which saves time during data collection. Because fewer organisms need to be identified and counted, it requires less effort. It is also a practical choice when there is limited time, equipment, or manpower. However, there are several disadvantages. A smaller quadrat covers only a small area, so it may not represent the entire habitat. It may also miss some species, especially those that are rare or unevenly distributed. As a result, it has a higher sampling error since the result depend heavily on where the quadrat is placed. Finally, it provides less accurate estimate of population density and biodiversity because the sample size is smaller. In conclusion, both larger and smaller quadrat have their own disadvantage and advantage. A large quadrat provides more accurate and representative data but require more time, effort and resources. In contrast, a smaller quadrat is faster and easier to use but may not accurately represent the habitat due to its limited sampling area. Therefore, the choice of quadrat size should depend on the objective of the study, the characteristic of the habitat, and the available time and resources. Thank you.
Okay, today I present question number 4, which is about the challenges ecologists face when estimating the distribution and abundance of organisms. Our practical was conducted at Taman Botani Putrajaya using the quadrat sampling method. Even though Taman Botani Putrajaya is not a forest, it is a large study area with different habitat conditions. Because of this, ecologists may face several challenges during sampling. There are 3 main issues. Difficulty in detecting organisms. Second, large and heterogeneous study areas. And third is sampling bias and limited accessibility. Okay, next slide is for our practical. We conducted the sampling at Taman Botani Putrajaya using a 6 meter by 6 meter quadrat with a total area at 36 square meters. Inside the quadrat, we identified and conducted all plant species and calculated their population density. Our group recorded 5 plant species. Although this method helps estimate and distribution and abundance of plants, there are several challenges that can affect an accuracy of the result. Okay, next issue is difficulty in detecting organism. The first challenges is during our observation, some plants were difficult to detect because they were hidden by taller plants, grass, or leaf litter. Small seedlings can easily be overlooked during the sampling. If some individuals are missed, the total number recorded will be low and then the actual population. This causes an underestimation of population density and abundance. Okay, for next, my slide is large and heterogeneous studies areas. At Taman Botani Putrajaya is not a natural forest. It contains different habitat types such as large tree areas, garden, grassland, and lakeside areas. These habitats have different environmental and conditions, including sunlight, soil moisture, and vegetation. As a result, plant species are not distributed evenly. Therefore, one quadrat cannot represent the entire studies area, and multiple quadrats are needed to obtain a more accurate result. Okay, for next slides, the third challenges is sampling bias and limited accessibility. Some areas at Taman Botani Putrajaya, such as dense vegetation, lakeside, or protected gardens, may be difficult to sample. Researchers may choose only areas that are easy to access, such as a near pathway. They created sampling bias, where the sample does not represent the entire study area. To reduce this problem, quadrats should be placed in different representative locations. Okay, for last slide is the conclusion. The 3 main challenges can affect an accuracy of population density estimates. Therefore, using the representative sampling methods such as placing quadrat in different habitat types is important to obtain reliable ecological data.
Next slide is practical 2 question 5. What is the difference in the number of plant species in the quadrant? Hypothesize or make your own assumption. Discuss the factor that will have caused this type of distribution. The number of plant species differs among quadrant because plants are not distributed uniformly within a habitat. Their distribution is influenced by environmental condition, biological interaction, and disturbance, resulting in variation in species richness from one quadrant to another. One major factor is light availability. Quadrant areas receiving greater sunlight generally support a higher diversity of plant species because sunlight is essential for photosynthesis, whereas heavily shaded areas beneath dense vegetation often contain fewer species. Second, soil characteristics including nutrient availability, pH, moisture, and texture also influence plant growth. Fertile soils provide better conditions for plant establishment and support a greater variety of species. In addition, water availability plays an important role as moist areas can support more species than dry habitats where only drought-tolerant plants can survive. Competition among plants for light, nutrients, water, and space may also reduce species diversity when dominant species outcompete others. Furthermore, difference in seed dispersal by wind, water, or animals can cause certain areas to receive more seeds, leading to higher plant abundance and diversity. Finally, human activities and natural disturbance such as land clearing, flooding, storms, or fire may alter vegetation patterns and reduce species diversity. Natural variation in plant distribution also contributes to difference among quadrants, as some species occur in cluster while others are randomly distributed. Based on our hypothesis, the difference in plant species among quadrants are mainly caused by variation in environmental conditions, resource availability, competition, and seed dispersal. Therefore, we expect quadrants with more favorable conditions to have a higher diversity of plant species compared to quadrant with less favorable conditions.